Hi, my name is Raymond. I usually go by Ray, but because I wrote so many emails since I was a grad student, I just shortened my name. So today we'll talk about applications of GIS in Arctic permafrost hydrology and biogeochemistry. Okay. So most of this work comes from my postdoc at BYU. Um, Due to grad school and different jobs, I had to go to a lot of different places. I studied economics and philosophy at UC Davis. I got a master's in geography at San Diego State and a PhD in hydrology and soil science at Virginia Tech. I had a postdoc at Pacific Northwest National Lab where we did numerical modeling of rivers. So basically just go into an office every day and write codes for 8 hours and then go home. And then I took a postdoc at BYU. Most of our work was in the area at Utah Lake, Great Salt Lake, if you know the area. But we also had field work in Alaska, Arctic Alaska. So due to the coronavirus at the time, we could not afford to send people up there for brief visits, for like a 1 week or 2 week visit, because every visit required the project to pay for a quarantine hotel for 2 weeks and food and all that. So I went up there for the whole summer, about 100 days, and brought an undergrad student with me. After that, which I'll talk about in a minute. After that, I took a visiting professor position at Washington College, which is a small liberal arts college in Maryland. It's the oldest small liberal arts college in the country. And I developed a course on the Arctic environment while I was there. And now I made the superior move to Wisconsin, and I'm a new assistant professor of GIS there. So I head the GIS program, and I thought I would talk a little bit about the ongoing research I have in the Arctic, and then just share a few thoughts on GIS in education and welcome feedback, because a lot of the students will eventually end up working for some of you guys. All right, so the main question that we had with Arctic permafrost hydrology and biogeochemistry is, how is matter transported across Arctic landscapes? It's not immediately clear. And we broke that down into smaller questions, so I'll just read them out. What type, quantity, quality of material is moving hydrologically across vast river basins? Typically, we focus on carbon and nutrients. How does this material get added, transported, and transformed along the river network? What is the contribution from terrestrial and aquatic environments? Are exogenous materials entering the basin? These are a hot topic. Typically, we look at mercury and microplastics. And then how are these novel materials getting added, transported, and transformed? These were our guiding questions. We started from this basic premise that river basins are vast and unregulated in Arctic Alaska. These are just some photos from the air. By vast, I mean they're humongous. There are no people, there are no roads. They're unregulated, hydrologically speaking, so there are no dams, there are no reservoirs. There are just natural rivers that flow to the Arctic Ocean. Quite unusual in the lower 48. We don't see that anymore. And the other thing that was driving our work was the distinct hydroclimate season there. So it's a very unusual place. The seasons are very different from what we're used to. Water flows for 3 months of the year. So there's some photos up there of flowing water. That's what it looks like throughout the summer hydrologic season, those 3 months. And those are in the vertical line, the colored vertical lines at the bottom. The landscape is frozen for the remainder of the year, so about 9 months, the ground is frozen. It's not just the water, but the ground, the soil, the rock, all of that. So typically you would sample somewhere between June and September. And importantly, the warm season is coincidental with other processes. So it's warm in the summer. The streams are also flowing in the summer. There's vegetation growth, and that's when there's the highest microbial activity. In the middle here, there's just some soil temperature profiles. There's a dashed vertical line that indicates 0. So left of the dashed line would be frozen soils, and right of the dashed line would be thawed soils. And when you have thawed soils, then you get water moving through the soil system. When it's frozen, the water runs straight off the surface. So when I first got to BYU, they had done some field work, and my advisor at the time, his name was Ben Abbott, he asked me to write up the results. And so we did, and we published this paper. In doing this, we took the field data and we did a stream network analysis. So we used GIS to, to delineate the watersheds, but also calculate the stream orders. So you can see here in this panel B, C, and D, those are the 3 main watersheds that we were studying. In the left panel A, there's a blue symbol inside an orange symbol. That's the field site. I mean, the field camp, the field station, Tulik Field Station. And it's off of the one road that goes north from Fairbanks to the Arctic Ocean. So we stayed in the field station and then sampled the 3 watersheds. The sampling for this paper was done by previous students. And then in the panels B, C, and D, you see increasing stream orders represented by different colors. So as the streams converged, the streams got bigger until it got to the main stem, which is in the yellow lines in those panels B, C, and D. So we did a stream network analysis showing how there is hydrologic dispersal of microbes in permafrost watersheds. Bacterial plankton communities were distinct by season, landscape type, sediment association, and stream order. That means that you have different communities of bacteria depending on where you were in the watershed. Sediment association just means that the bacteria are attached to some sediment, and sediment are considered to be migrants. They move with flow. If the bacteria are not associated with sediment, that means that they're likely to be natives of that area. The bacteria have reproduced, and you have new bacteria that are not associated with sediment. So it's really important whether the bacterial plankton were moving or not, migrants or not, if they came from soil systems upslope or not. Higher bacterial diversity with sediment-associated soil-derived migrants in early season in lower stream orders. You can see here the stream order on the x-axis and species diversity on the y-axis, and there's a clear pattern as you increase in stream order. So you get a picture of how these bacterial plankton are moving through these watersheds. There's also biogeochemical function that is dependent on microbial community composition in distinct landscapes. It matters what the community composition is because it affects the biogeochemistry of those places. It's not just where are the bacterial plankton, it's what are they doing. So there was a lot of biogeochemical activity in terms of iron up here in the alpine site, in the mountain site, in the green one, chloride in the tundra site, which is the blue one, and phosphorus, SRP, soluble reactive phosphorus, in the tundra one, punctuated by lakes and ponds. So in the orange watershed, it's similar to the blue one. They're both tundra vegetation, but in the orange watershed, the stream network is interrupted by ponds and lakes. So because of this different topography, you definitely see the differences in the dispersal of bacterial plankton, but also the biogeochemical function of them. And here is a significant trend between phosphorus processing, and species richness, especially in the orange watershed, which is the lake watershed. So we use GIS to identify where the hotspots were of this biogeochemical functioning and how it was related to species richness in the three watersheds. But we also knew that transport of material depended on other ways to break down the stream form. So you can look at the stream order, which is say like from 1 to 10 as the streams converge. But there were distinct forms like water tracks. That's a very Arctic type of stream. It's pictured here on the top row in different basins. It is considered to be a zero order stream. It is not in a defined stream channel. It's the closest we get to soil water, but it is basically surface runoff. And then we looked at tributaries, which are larger streams, and the main stem of the river. The stream order didn't really help us identify these three categories. So we thought about it a little differently. There's also the ecological province. So if you look at the left side, you've got the alpine, upper foothills, lower foothills, and coastal plain. And we thought it was very important to break the landscape up into these ecological provinces. They have different landscape positions. They have different topography and slope, which affect the speed of flow or velocity of flow. They have different vegetation types. And of course, they have different microbial communities. And then there's different glaciation histories across the landscape, which affects soil texture and soil chemistry. So we wanted to scale up from the mesoscale watersheds to larger river basins, something like this. This map is flipped upside down with north pointing to the bottom of the map. In consultation with my advisor at the time, we came up with a new experimental design. So we sampled along the longitudinal direction of flow. So we thought about where water came from. It came from hill slopes in these water tracks, and that water would eventually run into a tributary, and that tributary would eventually run into the main stem, and the main stem would eventually run into or flow into the Arctic Ocean. So we took samples along that flow direction. We collected samples of water and soil at clusters of sites, and each cluster had sites with different stream forms. So imagine we flew a helicopter, we landed somewhere, and we took a sample in a water track. We looked around us, we took a sample at a tributary, let's say 5 minutes walk, and then we took a sample at a main stem site. These clusters were at different ecological provinces and in multiple river basins. And we sampled at the beginning and end of hydrologic season to capture the change. We did additional sampling in the Sagavanirktok Basin because the SAG runs along the gravel road, so we can go anytime. And we analyzed the soil and water for physicochemistry, or the stream for physicochemistry, carbon, nutrients, mercury, and microplastics. The problem was I was a new postdoc, and I had an undergrad student who was brand new as well. I'd never been in the Arctic, and we didn't really know what we were looking at, and we couldn't know what we were looking at because we were in a quarantine hotel for 2 weeks. And the hotel was in Fairbanks. So we were just going by what we saw in a GIS program and emails from our advisor. So Ben sent this Google Earth file. We looked at it, and he said, well, you guys got to look for some sites that follow all of these rules. So I'll just read them out. The sites have to occur in positions on the longitudinal profile. They have to have an appropriate watershed size for the stream form. So the water track site should have a small watershed. That would make sense. Watersheds are representative of the ecological province. The sites within the clusters are not too far on foot because we have to be able to land somewhere and run around and get all the samples and run back to the helicopter in a short amount of time to get to the next site. And there should be no major disturbances visible from space. So no off ice, which is like a frozen stream feature. No thermal karst, which is like a slump in the landscape due to permafrost thaw. And you should not be influenced by the haul road, which is the one gravel road. He also said in the email, well, he was a busy guy, so he didn't get to us until the day before we were to go out for the first time. And he said, for the sampling tomorrow, this is just verbatim from the email. He says, for the sampling tomorrow, there are a few sites where you will park on the side of the haul road itself. In other words, there aren't turnoffs. This is fine, but you do need to be careful about not sliding off the road while also getting far enough off to not cause problems for others on the road. We didn't really know what to expect. The road just has truck traffic. So we have a big SUV, but it's quite small compared to the trucks. And I don't know if you can tell, but there's nothing out there. There's nobody out there. There are no landmarks out there. There are no crossroads out there. There's just nothing. So we had to be very sure of the sites that we picked because we had one shot and we were paying a lot of money for this helicopter, for the pilot, for the fuel. There was a packed helicopter schedule for everyone in the camp. We had our days picked out and we had to have a flight plan that I had to write up. And we were just going out into the middle of wherever that neither of us, Jansen, the student, nor I had known before. So we had to be very careful in our planning. So in the quarantine hotel, we got together and Jansen looked at the sites on Google Earth and QGIS. And we just had to make very, very, very sure of our coordinates because we had to get them to the pilot in advance so he could plot a plan to get us to all the places we needed to go. And the pilot was like a taxi driver. He did not care. It was not his problem. He was just flying us he was like a shuttle driver, just getting us to wherever we needed to go. So it was up to us, or I guess me, to kind of make sure that we we're going to the right spots. These watersheds had to be delineated correctly. It was very tricky with the water tracks because they are such small features. So here's the actual day that we went out. We went out about 7 in the morning to 9 at night. But at the time, it was summer, so it was 24-hour sun. It didn't really matter. That's when we came back on the left side at 9 o'clock at night. This is one of the sites we went to and landed the helicopter. We had to identify the water track visually quickly to make sure that that's where we wanted to sample. That would be the tributary. And we had to run around back and forth, pick up bottles, drop off bottles. And we did that, I can't remember how many times, like 12 times. All right, so what did we find? These are some preliminary results. We did find some patterns across stream forms. There was production and material addition in the terrestrial environment. You can see it with the high DOC concentrations on the left in the water tracks, decreasing toward the main stem. There were patterns across ecological provinces. You see material transformation in the aquatic environment in the red arrows. Interestingly, there was an abrupt change in direction between the lower foothills and the coastal plain. And there were few patterns across river basins. Everything was flat across river basins, which suggests that glaciation might not be as big of a deal. And it's the previous two or the former two factors that are more important in controlling stream water chemistry. There were distinct biological signals in the water tracks. There were signals that decreased along the stream gradient. Here on the left panel, you have dissolved oxygen in percent. All the sites were saturated at about 100%, but the water tracks were not. The water tracks are in red and indicated that there was high consumption of oxygen and likely high microbial activity. Also in the water tracks, there was high DOC, correlated with high chlorophyll, and nitrate was limiting. And we started to do some spectral analyses, looking at wavelengths of light, and that helped give us a sense of the quality of the organic matter. And it indicated to us that the streams and the water tracks, or the water tracks, had material that was highly aromatic. But we know that light is also a dominant decomposer in the Arctic. It's not just the microbes. Sometimes it's an abiotic thing, just natural light breaks down matter. Microbial activity and light availability are highly seasonal and in phase, so they're confounding and it's a little bit complicated. There are distinct microbial community compositions and function along the stream network. There is the puzzle of whether bacterial plankton are dispersed hydrologically or selected due to, say, there are suboptimal living conditions for bacteria and they die off. So there is a change in the community composition due to the environment, not due to the dispersal. There are distinct light regimes along the stream network. It's dark in the water tracks versus light in open water. And these are clean oligotrophic waters, which allow light to penetrate streams, ponds, and lakes. Ben was pretty clever. We used the water samples that we had collected, and we appropriated them for a new experiment that the student would do. Here's Jansen, the undergrad. He further filtered them to remove the confounding bacteria and sediment, and then he inoculated them with raw sample water that contained bacteria. And then he incubated these samples in light and dark treatments in a mesocosm. And by mesocosm, I mean he handmade some boxes. Some had opaque covers and some had transparent mesh covers. And he put them in the lake, in Toolik Lake, so we could see them from our lab facility. And he monitored them and analyzed them for DOC, nitrate, and turbidity every few weeks. Eventually, he presented his results at AGU, which is the annual meeting of the American Geophysical Union, showing that photodegradation dominates DOC mineralization across watershed sizes and ecosystem types. But I wanted to scale up further from river basins to a statewide north-south transect across permafrost zones. When I left the camp at the end of the summer, it was September, it was the end of the hydrologic flow season. Ben was gracious enough to let me take my time coming back. So I took 2 weeks to get back to Utah, and I drove across the state of Alaska. And every time I crossed the river, I took a sample. Ended up taking over 100 samples. And then I also decided to fly out to the Aleutians and sample everything there as well. This is just what a typical stream looks like. After you see so many streams, they all kind of look the same. The study design includes multiple large river basins. It also includes glacial inputs. And it includes the Aleutian Islands. I don't think there's been much hydrologic research done there. It's Dutch Harbor in Unalaska. Interestingly, the vegetation there was tundra vegetation, which is similar to what you would see in the Arctic as well. All right, so just want to share a little bit of the preliminary analysis. This is the workflow that we came up with. I had some partners at different universities. I think also the National Park Service and the USGS. So we wanted to map the sample sites and characterize the stream water chemistry data that we had in hand, and then use GIS to delineate the watersheds, summarize state factors, ultimately to make empirical models of stream and river chemistry in the late summer. This was the deepest thaw of the seasonally frozen active layer. This is when the most water would go through the soil system and deliver material into the streams and rivers. If there's a change in permafrost coverage in the next few years or decades, then this would give us a sense of what we might expect in terms of stream water chemistry. So here is a map of all the sites across Alaska in blue and the watersheds. Here's the data that we had. We got nitrate at the top, DOC on the bottom. And these were the state factors that we looked at: topography, elevation, aspect, slope, potential incident radiation, and topographic position index. We looked at catchment area and stream length, climate, soils, so permafrost extent, soil organic carbon, soil temperature, depth to the active layer, parent material and glacial history, land use and cover, and burn extent and severity. All of these were acquired from outside, and they're just large shape files that covered the whole state. So this is from our partner at the USGS. He put this together. He used a digital elevation model that had 5-meter resolution. He used some algorithms to fill in the sinks, calculate flow direction and flow accumulation, to delineate the watersheds, and then come up with some statistics. He generated a correlation matrix, and we start to see some patterns suggesting that permafrost extent and catchment slope are likely drivers of fluvial biogeochemistry, especially as it relates to DOC. This is pretty fresh. I just heard from him a few days ago, so we're still, I guess, analyzing this even further. This is kind of the first pass. So that's all I have for you on the Arctic. I also wanted to use this time to introduce myself. I'm a new professor here. I just moved here. I don't know anyone here. I just know my wife. That's about it. And we just stay at home all the time because we're doing some renovations. So I don't know anyone here, and I just wanted to meet you guys and let you know that I'm in charge of the GIS program at University of Wisconsin-Superior, and kind of describe the program, describe some of the changes that I've been planning, and maybe get some feedback if you have some suggestions on how to improve it. So just an overview. The program currently consists primarily of 3 GIS courses. I think of them as GIS 1, 2, and 3. They have specific designations: GEOG 241, 343, 442. We don't have a remote sensing course. We don't have a cartography course. We don't have a data visualization course. This is pretty much it. So I've been trying to think about how to put some of those other elements into these 3 courses. I've been thinking that there are actually many ways to improve GIS in education, starting with the information systems side, trying to increase technical skills in both courses and mentorship. And what I mean by that is teaching ArcGIS Pro, QGIS, R, and potentially numerical modeling. This is the GIS 1 course. And early on, we have a lab activity, corresponding activities back to back, one in ArcGIS Pro and one in QGIS, just to broaden the perspectives of the professional opportunities available to the students after they complete the program. I was at a national lab. We were swimming in the money. But for some reason, we always used QGIS because it was free. So, when I was a student, everything was in ArcGIS. So I think it's good for the students to kind of see both sides of it because you never know where you're going to end up and what they're using. In the second semester, if they decide to continue, then we'll go over some additional software that places GIS into larger workflows so they understand where the data are coming from and what to do with them afterward. And there's a big shift toward open science and scripting and publication of data sets and publication of codes and workflows themselves. So there are several lab assignments that go into our statistics and also Jupyter notebooks. And then finally, the third course will assume that the student has these technical skills, and we do not need to practice the technical skills per se, but we can combine the technical skills and critical thinking to solve complex problems. And these problems range across the environmental disciplines and the social disciplines. I'm a big fan of the sphere words, the sphere model. You got the lithosphere, hydrosphere, biosphere. Everybody knows these words. Atmosphere. There are new words that people have made up, the anthroposphere, the technosphere. 4 out of, I think, 23 of the students in my GIS 1 class are from the Department of Transportation and Logistics. And so the technosphere deals with, say, road networks or subway networks. So each week, the students will have to solve a problem in a different place on Earth. And this allows them to practice solving problems in a place that they might not be comfortable with or familiar with, just to demonstrate and develop their independence. And I'm developing a new course on R. We're going to roll that out next semester. It's entirely on R. Ranges from data wrangling to data visualization to interpretation, finishing up with a debate, base R versus the tidyverse. I think people tend to pick one or the other. R gives students powerful tools to analyze data that are read into a GIS or derived from a GIS. So it's really important to know what you are doing before GIS and what you're going to do after GIS. I think there is a debate about whether undergraduate students can handle R, if it's too much, if it's too little, if it's important for them. So we should try to teach it, or if we're just discouraging them. I don't know. But undergraduate students wrote their own scripts in R for these research products. So Jansen wrote the scripts to make those figures. Tasha wrote the scripts, not just to make those figures, but she wrote all the scripts to analyze all the data. And I came in and I wrote the paper. And now she is a master's student with Byron Crump, a microbiologist in University of Oregon. Jansen's finishing up his undergrad at BYU. And I've worked with many other students at BYU who all wrote their own scripts. So hopefully it works out. I'll give it a try. And there are always needs for numerical modeling in the Arctic. So if any student wants to take on a separate research project, we'll be glad to help them out. This is just the DEM of those 3 study watersheds, and then the DEMs or the watersheds represented in a mesh so that you can introduce it to a numerical model. And then going to the geographic side, I think it's important to increase the understanding of the meanings of words, concepts, and places. I think a lot of times we take them for granted, and it's important because we're mapping words or we're mapping places. So I encourage my students to read broadly. What I mean by that is read meta-analyses of studies of concepts, read national policy documents, just different types of documents. And then I also try to incorporate multiple data sets to understand a word, concept, or place. And by that, I mean, I put music into the courses, I put songs with lyrics into the courses, and sometimes sonifications, which are distinct. That's just when you convert data to an audio format. There's a lot of talk about visualization, so I try to think about sonifications as well. And then sometimes I show some antique maps and we kind of discuss what they mean. This all started when I was working on the critical zone. I don't know if anybody here knows or has heard of the critical zone, but it's a word that was coined by the National Research Council in 2001, and it spurred a lot of research on earth science. It's a new term based on old ideas. And when they introduced it in this document in 2001, it was interesting because they defined it in a way that was contradictory in some ways, or there was tension between the different ways that they defined it. So they had a spatial definition here because they described the critical zone as being the land surface and its canopy of vegetation, rivers, lakes, and shallow seas. And the spatial definition had an area. It was the terrestrial surface of the planet, and it had a height. And that height varied at a given location and at multiple temporal scales, because depending on whether a tree existed at that time and the height of that tree top, you would have a different size of the critical zone at any given time. There was also a process definition because in the same sentence, they go on to say that the critical zone is where processes occur that determine the availability of nearly every life-sustaining resource. And in this way, people have latched onto this meaning as well and said that, oh, their study site is a critical zone in itself. It is not the area of the whole terrestrial land surface of Earth. It is more spatially limited. It is, say, a single watershed or basin. It can be an abstract definition, it can be just an ecosystem, and it can be physically agnostic. So it could be something like political boundaries, maybe a nation or so. So it's confusing when you talk about the critical zone, where are you pointing to? Everybody's thinking about a different place. Eventually, other researchers started to define the critical zone by some unifying thread or theme, like a material zone, say like the soil. The critical zone is defined as the soil. And then other people compete and say, well, maybe it's the trees. The trees control everything, so it's the trees. Or it's some biogeochemical cycles or hydrologic cycle or information flows or other things. And depending on what you are focusing on or emphasizing, the critical zone is going to look different to you. This is just a sample of the different conceptual models that people have used in their papers about the critical zone, and they will magnify different parts of the landscape. So it was pretty confusing. I read a bunch of papers about the critical zone and thought about it a little more, published this paper with some co-authors on the meanings of the critical zone. And we found that human activity was not limited to degrading the critical zone, but it also includes participating in cultural life, because there were papers that talked specifically about how the critical zone was the place where we build our shelters, grow our food, extract our minerals, educate our children, govern our societies, run our economies, and leave our legacies. Basically, the critical zone was our home. One thing I want to point out is, definitions of words matter. There are other critical zones in other places, depending on the discipline that you are in. And if you are thinking about a field-based science, we should all be kind of clear on terminology because you're referring people to specific places on Earth. For the GIS1 class, I just went over this the other day. How can you make a map of pond ecosystems in the Great Plains? It can be quite confusing, the definition of each of these single words. The maps, ponds, ecosystems, and Great Plains. Here there's a map of 50 different authors drawing different boundaries around the Great Plains. There's a review paper here where the author reviewed 321 definitions of the word map. No consensus. Here, a team analyzed 519 papers with 54 definitions of pond. Again, no consensus. Ecosystem was coined by Arthur Tansley in 1935. There have been many, many, many changes to the word ecosystem. It is a ubiquitous word. Everybody uses it and knows it, but we all define it differently, depending on what emphasis we put on it. And then the Great Plains, an analysis of 50 maps of the Great Plains region, no consensus. So it's really important for a GIS student to understand exactly what they're looking at, exactly what they're trying to map. Also, there have been many changes to U.S. national Arctic policy for complex socio-ecological reasons. When we try to map the Arctic, or when we think about the Arctic as a region, where are we drawing the boundaries? Is it a latitudinal boundary? Is it the watersheds or the basins that flow into the Arctic Ocean? How do we draw those boundaries? Is it the Arctic Council, the nations that have land above the Arctic latitude? And how do we approach it as a nation? We use different documents. Here you have a classified or formerly classified secret memo written by the National Security Council in the 1970s. And here you have something that looks more like a PR brochure that is a public document. I also play music. This is not really music per se. It's a commercial. It's a Toyota commercial for Tundra trucks. But it shows the truck in different situations. And it goes on, another commercial. And I asked the students, is this the Tundra or not? And they always, or they have said correctly that it is not. But it gets a little more complicated. Maybe I can start here. This one's a little more beautiful. It is about the tundra. And it's filmed in the mountains with snow. It's the BYU Women's Chorus. But it's not the tundra. It's filmed in Utah. So it's good to know. It's good to assess. It's good to evaluate the documents and the evidence in front of you. And then I play this sometimes. It's Thriller, Michael Jackson. This was filmed in the research station at Tulip Field Station in the actual tundra, just to give students an idea of what the tundra actually looks like. So, and then here are some antique maps that we look at, maps of the world when California was an island or the Arctic. Finally, the last thing, try to promote authentic attachment to projects and study areas, keep them interested. For example, student-led creative endeavors such as branding. What do I mean by that? When Jansen and I were stuck in the quarantine hotel, we thought, hey, it started like this. I thought we had these bug shirts and they are jackets and there's a zipper and it covers your whole face. But you really need a hat or visor to keep the jacket out of your face. And we had not thought of that. So I offered to buy Jansen a visor, and I thought it would be interesting to customize the message on the visor, like put his name on it. And he told me that he did not like visors. So we thought, okay, let's make a baseball cap. And we started to think, hey, maybe we'll make a logo to put on the baseball cap. So we started to think about it, and I realized that the second letter and the penultimate or second to last letter was an O, and that could be replaced with the sun. And these are common photos of the sun where you see that the sun does not set below the horizon because it's 24-hour sun in the Arctic. So we started to come up with a design, and he had all the skills on the computer to put this together. And we finished with this. Eventually, we played with ideas for creating merchandise, started to put our logo in different places. And then we advertised the Tulik hat around camp, put it on the bulletin board in the cafeteria. We put it on the slide reel on the TV in the cafeteria. And we started an order form. And it surprised us, but a lot of people ordered the hat. Eventually, the hat came to camp, and it was quite popular. Finally, the two-leg hat got its own Twitter page. So that's all I have for you. Thank you.